the hallmarks of the scientific method is that it depends on empirical data. To be a proper scientific investigation, the data must be collected systematically. However, scientific investigation does not necessarily require experimentation in the sense of manipulating variables and observing the results. Observational studies in the fields of astronomy, developmental psychology, and ethology are common and provide valuable scientific information. Theories and explanations are very important in science. Theories in science can never be proved, since one can never be 100% certain that a new empirical finding inconsistent with the theory will never be found. Scientific theories must be potentially disconfirmable. If a theory can accommodate all possible results, then it is not a scientific theory. Therefore, a scientific theory should lead to testable hypotheses. If a hypothesis is disconfirmed, then the theory from which the hypothesis was deduced is incorrect. For example, the secondary reinforcement theory of attachment states that an infant becomes attached to its parent by means of a pairing of the parent with the primary reinforcer, food. It is through this secondary reinforcement that the child-parent bond forms. The secondary reinforcement theory has been disconfirmed by numerous experiments. Perhaps the most notable is one in which infant monkeys were fed by one surrogate wire mother, while a surrogate cloth mother was available. The infant monkeys formed no attachment to the wire monkeys and frequently clung to the cloth mothers. If a hypothesis derived from a theory is confirmed, then the theory has survived a test, and it becomes more useful and better thought of by the researchers in the field. A theory is not confirmed when correct hypotheses are derived from it. A key difference between scientific explanations and faith-based explanations is simply that faith-based explanations are based on faith and do not need to be testable. This does not mean that an explanation that cannot be tested is incorrect in some cosmic sense. It just means that it is not a scientific explanation. The method of investigation in which a hypothesis is developed from a theory and then confirmed or disconfirmed involves deductive reasoning. However, deductive reasoning does not explain where the theory came from in the first place. In general, a theory is developed by a scientist who is aware of many empirical findings on the topic of interest. Then, through a generally poorly understood process called induction, the scientist develops a way to explain all or most of the findings within a relatively simple framework or theory. An important attribute of a good scientific theory is that it is parsimonious. That is, that it is simple in the sense that it uses relatively few constructs to explain many empirical findings. A theory that is so complex that it has as many assumptions as it has predictions is not very valuable. Although strictly speaking, disconfirming a hypothesis deduced from a theory disconfirms the theory, it rarely leads to the abandonment of the theory in the field. Instead, the theory will probably be modified to accommodate the inconsistent finding. If the theory has to be modified over and over to accommodate new findings, the theory generally becomes less and less parsimonious. This can lead to discontent with the theory and the search for a new theory. If a new theory is developed that can explain the same facts in a more parsimonious way, then the new theory will eventually supersede the old theory. It is important to keep in mind that sampling bias refers to the method of sampling, not to the sample itself. There is no guarantee that random sampling will result in a sample representative of the population, just as not every sample obtained using a biased sampling method will be greatly non-representative of the population. Imagine a university newspaper ran an ad asking for students to volunteer for a study in which intimate details of their sex lives would be discussed. Clearly, the sample of students who would volunteer for such a study would not be representative of the students at the university. Similarly, an online survey about computer use is likely to attract people more interested in technology than is typical. In both of these examples, people who self-select themselves for the experiment are likely to differ in important ways all differences were chance differences. Independent variables often have several levels. For example, in the smiles and leniency case study, the independent variable is type of smile, and there are 4 levels of this independent variable. 1. False smile, 2. Felt smile, 3. Miserable smile, and 4. Neutral control. Keep in mind that although there are 4 levels, there is only one independent variable. Designs with more than one independent variable are considered next. In the bias against associates of the obese experiment, the qualifications of potential job applicants were judged. Each applicant was accompanied by an associate. The experiment had 2 independent variables: the weight of the associate, obese or average, and the applicant's relationship to the associate, girlfriend or acquaintance. This design can be described as an associate's weight, 2, by associate's relationship, 2, factorial design. The numbers in parentheses represent the number of levels of the independent variable. The design was a factorial design because all 4 combinations of associate's weight and associate's relationship were included. The dependent variable was a rating of the applicant's qualifications on a 9-point scale. If 2 separate experiments had been conducted, one to test the effect of associate's weight and one to test the effect of associate's relationship, then there would be no way to assess whether the effect of associate's weight depended on the associate's relationship. One might imagine that the associate's weight would have a larger effect if the associate were a girlfriend, rather than merely an acquaintance. A factorial design allows this question to be addressed. When the effect of one variable does differ depending on the level of the other variable, then it is said that there is an interaction between the variables. Factorial designs can have 3 or more independent variables. In order to be between subjects designs, there must be a separate group of subjects for each combination of the levels of the independent variables. A within-subjects design differs from a between-subjects design, in that the same subjects perform at all levels of the independent variable. For example, consider the ADHD treatment case study. In this experiment, subjects diagnosed as having attention deficit hyperactivity disorder were each tested on a delay of gratification task after receiving methylphenidate, or MPH. All subjects were tested 4 times, once after receiving one of the 4 doses. Since each subject was tested under each of the 4 levels of the independent variable dose, the design is a within-subjects design, and dose is a within-subjects variable. Within-subjects designs are sometimes called repeated measures designs. In a within-subjects design, it is important not to confound the order in which a task is performed and the experimental treatment. For example, consider the problem that would have occurred if, in the ADHD study, every subject had received the doses in the same order, starting with the lowest and continuing to the highest. It is not unlikely that experience with the delay of gratification task would have an effect. If practice on this task leads to better performance, then it would appear that higher doses cause the better performance when, in fact, it was the practice that caused the better performance. One way to address this problem is to counterbalance the order of presentations. In other words, subjects would be given the doses in different orders in such a way that each dose was given in each sequential position an equal number of times. An example of counterbalancing is shown in Table 1. It should be kept in mind that counterbalancing is not a satisfactory solution if there are complex dependencies between which treatment precedes which and the dependent variable. In these cases, it is usually better to use a between-subjects design than a within-subjects design. An advantage of within-subjects designs is that individual differences in subjects' overall levels of performance are controlled. This is important because subjects invariably will differ greatly from one another. In an experiment on problem solving, some subjects will be better than others regardless of the condition they are in. Similarly, in a study of blood pressure, some subjects will have higher blood pressure than others regardless of the condition. within subjects' designs control these individual differences by comparing the scores of a subject in one condition to the scores of the same subject in other conditions. In this sense, each subject serves as his or her own control. This typically gives within subjects' designs considerably more power than between subjects' designs. That is, this makes within subjects' designs more able to detect effects of independent variables than are between subjects' designs. Within subjects' designs are often called repeated measures designs, since repeated measurements are taken for each subject. Similarly, within subjects' variables can be called repeated measures factors. Designs can contain combinations of between subject and within subject variables. For example, the weapons and aggression case study has one between subject variable, gender, and two within subject variables, the type of priming word and the type of word to be responded to. The concept of causation is a complex one in the philosophy of science. Since a full coverage of this topic is well beyond the scope of this text, we focus on two specific topics. 1. The establishment of causation in experiments, and 2. The establishment of causation in non-experimental designs. Consider a simple experiment in which subjects are sampled randomly from a population and then assigned randomly to either the experimental group or the control group. Assume the means for the dependent variable differ between the groups. Does this mean the treatment caused the difference? To make this discussion more concrete, assume that the experimental group received a drug for insomnia, the control group received a placebo, and the dependent variable was the number of minutes the subject slept that night. An obvious obstacle to inferring causality is that there are many unmeasured variables that affect how many hours someone sleeps. Among them are how much stress the person is under, physiological and genetic factors, how much caffeine they consumed, how much sleep they got the night before, etc. Perhaps differences between the groups on these factors is responsible for the difference in the number of minutes slept. At first blush, it might seem that the random assignment eliminates differences in unmeasured variables. However, this is not the case. Random assignment ensures that differences on unmeasured variables are chance differences. It does not ensure that there are no differences. Perhaps, by chance, many subjects in the control group were under high stress, and this stress made it more difficult to fall asleep. The fact that the greater stress in the control group was due to chance does not mean it could not be responsible for the difference between the control and the experimental groups. In other words, the observed difference in minutes slept could have been due to a chance difference between the control group and the experimental group, rather than due to the drug's effect. This problem seems intractable since, by definition, it is impossible to measure an unmeasured variable, just as it is impossible to measure and control all variables that affect the dependent variable. However, although it is impossible to assess the effect of any single unmeasured variable, it is possible to assess the combined effects of all unmeasured variables. Since everyone in a given condition is treated the same in the experiment, differences in their scores on the dependent variable must be due to the unmeasured variables. Therefore, a measure of the differences among the subjects within a condition is a measure of the sum total of the effects of the unmeasured variables. The most common measure of differences is the variance. By using the within condition variance to assess the effects of unmeasured variables, statistical methods determine the probability that these unmeasured variables could produce a difference between conditions as large or larger than the difference obtained in the experiment. If that probability is low, then it is inferred, that's why they call it inferential statistics, that the treatment had an effect and that the differences are not entirely due to chance. Of course, there is always some non-zero probability that the difference occurred by chance, so total certainty is not a possibility. It is almost a cliché that correlation does not mean causation. The main fallacy in inferring causation from correlation is called the third variable problem, and means that a third variable is responsible for the correlation between two other variables. An excellent example used by Lee in 1975 to illustrate this point is the positive correlation in Taiwan in the 1970s between the use of contraception and the number of electric appliances in one's house. Of course, using contraception does not induce you to buy electrical appliances, or vice versa. Instead, the third variable of education level affects both. Does the possibility of a third variable problem make it impossible to draw causal inferences without doing an experiment? One common approach is to simply assume that you do not have a third variable problem. This approach is not very satisfactory. However, be aware the assumption of no third variable problem may be hidden behind a complex causal model that contains sophisticated and elegant mathematics. A better, though admittedly more difficult, approach is to find converging evidence. This was the approach taken to conclude that smoking causes cancer. The analysis included converging evidence from retrospective studies, prospective studies, lab studies with animals, and theoretical understandings of cancer causes. A second problem is determining the direction of causality. A correlation between two variables does not indicate which variable is causing which. Sometimes this is a trivial problem, and sometimes it is not.